Well, thanks for inviting me to speak here. Thanks for waiting for the last talk, so last but not least, obviously. It's a great place to have a meeting here. I particularly like the fact that they have a blackboard in the toilet in case you get inspiration. But today I'm going to talk about design and control of microbial communities. Because I'm the last talk, I'm kind of going to talk about open challenges a little bit. I'm also going to talk about kind of future methods. Hopefully it's not too controversial, but we'll see. Okay. So my group works on a few different things. So we do modeling of cancer, mutational processes. So actually we model the dynamics of genomes, microbiome engineering, which is what I'll talk about in a minute. In the past, we've worked on gene regulatory networks, and we also work on biological computing. And in this part, we're interested in microbial communities and how they perform computations, but that's another talk. So what do I mean by microbiome engineering? So that's really kind of leveraging engineering tools and design principles to create or modulate host microbiome systems. So obviously, that's probably what a lot of you guys do as well. In terms of the human microbiota, this is already going on. So here's a defined fecal microbiota transplant in clinical trials. This is a single strain of Blautia. And this here is an engineered microbe that expresses an enzyme to stop this metabolic disease. So this is going on right now, and I'm coming slightly from the engineering biology or synthetic biology angle. So we've worked on different things here. So we've, microbial systems modeling. So that we've heard a lot about that today, and that's what I'll talk about mostly today. Synthetic microbial communities, bacterial biosensors, engineered probiotics, and we've also used worms as a host microbe model system, which is pretty cool. I could talk. I'm not going to talk about this too much, but we spent a lot of time on microbial community design. So in this paper, we looked at how to find systems that express certain dynamical properties, such as stable, steady state, or oscillations. This is using a kind of Bayesian inference. I'm not going to harp on too much about Bayes in this talk, but on another day I might do. Here we use the same Bayesian approach to search for chaotic communities, so bacterial communities. So this is theoretical work. We find that it's not uncommon that you can land on a chaotic system. And this one I'll talk a bit about now. This is one that we built. So this is work by Alex Fedorek, who's now a lecturer, or will be a lecturer at UCL, so that's cool. He developed this system called stabilized populations by community killing, which is a classic backronym that stands for SPOC, and you'll see our group has a habit of coming up with backronyms. So one thing I want to say is, in the engineering biology community, a SynCom, synthetic community, involves engineered bacteria or engineered microbes. You guys, you put together microbes, and then call that the synthetic SynCom. Okay, so just to point that difference out. Okay, so here we're trying to use E. coli and engineering the interactions in. So if you like, this is a bottom-up approach. So what we've been using is intercellular toxins called bacteriocins, which I'm sure many of you are familiar with. They're microbially produced antimicrobials. And this one that we use here is called microcin V. It's a very small peptide, and natively expressed by E. coli. So what we did here is we engineered one strain to express this bacteriocin, and we can do co-culture experiments, and we can show competitive exclusion and bacteriocin killing. So we can demonstrate that the bacteriocin can cause and create a niche for itself, which is what everybody suspects bacteriocins do. So then we engineered. I mean, it took a long time, and that's what the genetic system looks like. But we engineered this bacteriocin production with immunity, and the bacteriocin production is controlled by a quorum sensing molecule. Okay. So we've engineered kind of a negative feedback here, and this is how it works. So in this diagram here, the orange strains are the engineered ones, the blue ones are the native ones. Okay. So obviously, when you engineer a strain, it's weaker, it has lower fitness, so it starts to get out competed, and the number of quorum molecules goes down, which then induces killing of the native microbiota, which allows it to grow again, at which point we shut off killing, and it stops it overtaking the native microbiota. So that's the idea, and we have this like win-lose-win cycle. Okay, and as I said, Alex spent a lot of time building this, so it's on 2 plasmids. I won't go into the details, but it's there. So there was talk about flow cytometry earlier. So I just thought I'd mention this tool we also developed. So this is for quantitative flow cytometry. So we can calibrate the flow cytometer so that we have real units, which, for a physicist or chemist, that sounds ridiculous, I think, or an engineer, but often in biology we don't have real units, so it helps. And we can also normalize and calibrate the number of cells we have in the system. And if you're going to engineer things, then you need to know units, and you need to know that it's going to work the same somewhere else. So it's an important tool. If you're interested in using it, it's in R. So here's just some results of the system working. So what you can see here is that the co-culture establishes a 50-50 ratio, but we can also change the ratio via an inducer of arabinose. And we did that in plate reader. We also did it in this like pseudo chemostat mode, which is inoculations every few hours. Okay, so we do have some table benchtop bioreactors, but it's pretty challenging getting them to work in chemostat mode. But I'm sure you know that. So we're still working on that. So we extensively modeled this. So this is a mechanistic model. We didn't use Lotka-Volterra here, and fit the model and looked at, essentially this is kind of the engineering parameters of the system. So dilution rate on the x-axis and quorum molecule production rate. So this is AHL, and we can analyze this and look for the coexistence. And what we found was, if you can't, so leakiness is kind of how on it is when you try and turn it off, and that turns out to be really important. So we got about 10-fold change in bacteriocin production, but ideally we need about 100-fold, and that's kind of one of the challenges of engineering these systems. We also did a Bayesian approach to look at how robust this system was. And basically what you can see here is a measure of robustness of the system. We built this one here, which is the worst one out of these four. But at least we know that. And we're trying to build system 2, which is only slightly more complicated. So this is like a classic case of additional feedback loops giving more robustness to the system. So we're trying to build this as well at the moment, which would get us up here. So that would be good. So just to say, I think for you guys, I think you should think about engineering biology or synthetic biology to go and test some of your hypotheses, because if you go into your synthetic communities, your synthetic communities, and you get some hypotheses for the different interactions that are going on, and you get these system behaviors, you can recreate them in principle in E. coli, but it's pretty challenging. Okay, so now to the second part of my talk. So I know we're all bored of this now, okay, and we all go, no, it's all hype, and I was the same, okay, but I don't think we can turn our backs on AI and machine learning, okay, and I don't mean it's useful as a tool such as cell segmentation or identifying species in the flow, but I mean in a bit more deeper level. I think it's going to become a tool in the scientific method, and everybody's going to have to use it. So here's some examples. So, I'm not, these are all from Nature, most of them from DeepMind, but, it is making inroads into helping mathematicians, control of difficult systems, and I'll talk a bit about that. Particle physicists have been using neural networks for a long time, but we now have neural networks that you can build in physics and diffusion equations and Lotka-Volterra equations, things like that. You can use AI to give you insight into biological systems, okay? And I think that's where we should be going. So we shouldn't turn our backs. We should embrace it and try and make use of these systems, these approaches. So we've been working with reinforcement learning, which is arguably like the one that is really pushing forward these applications. Okay, AlphaFold wasn't reinforcement learning, but that's kind of an exception. So what is reinforcement learning? So with reinforcement learning, you have an agent and it interacts with the environment and it receives some reward. Okay, and the agent, given enough data, learns to maximize something called the return, okay, which is kind of like the integral of the reward. And we've been using model-free systems, which, it provides the advantage. So you don't have to have any assumptions on the underlying process. And this, this was the most famous recently. It wasn't that recent, but a few years ago, beating the Go champion, okay? So I'm going to talk a bit about two pieces of work, both done by Nathan in my group, first as a PhD student and then as a postdoc. And the first is in controlling microbial communities. And the second is using reinforcement learning for optimal experimental design, okay? Both of which I think you might be interested in, but I could be wrong. But okay, so what's the motivation for controlling co-cultures? So, if you put strains together, they're not guaranteed to coexist, right? And that might be okay when you're studying these communities, but maybe you want to improve some bioprocess or do some metabolic engineering, and you want to distribute metabolic load across different strains or species, okay? So there are challenges here: competitive exclusion, hard to predict long-term behavior, and establishing stability increases as the number of species increases. So we pose this as a problem that can be tackled with reinforcement learning. So in this simple example, we have two species. They compete for a common carbon source, so they're competing, and we assume a bioreactor, a chemostat environment. And then the goal is to keep the two species within a region in this state space, which is given by the number of each species. So you want to be in this green region. Okay. So to cut a long story short, we developed the agent. We can show that we can set some arbitrary levels on the numbers of the populations. I should say this is all computational work. We're still working on doing it experimentally, but the theory is there. So we can do this, and then we can get, this is like the policy, it's called, which tells you what the agent does. Oh, sorry, I forgot to tell you. These are both auxotrophs. In this case, they're auxotrophs for amino acids, but they could be different feedstocks. These are the inputs to the system, okay? So we after a long time, we can achieve almost perfect control, and here's the policy. So basically what this says is if N2 is high, then add the amino acid for N1. That's what it says. If you're down here, add the amino acid, add both amino acids. And if you're up here, it never explored that space. Okay, so that's how you interpret those. So we showed that this outperforms proportional integral control at long sampling intervals. And actually, I think it could be a really great way to control, for example, the number of cells in a turbidostat as well, because it can handle noisy pumps and noisy kits, so you could use the cheaper bioreactors. So I think it has lots of potential for that. So by itself, it can take a lot of data to train. Okay, and so what we showed is that if you had 5 bioreactors in parallel, you could train the agent in 24 hours. So you could train the agent to control the populations how you want by the end of the 24-hour period. So that's not too bad. I think most people who are running bioreactors can manage 24 hours. I mean, we've seen hundreds of days. So I think it's a good way to do this control. And it can actually do more complex things, and we haven't really explored what this can do because we've been working on the second part heavily, but it can do things that normal control can't. So for example, say you have a microbial product that was formed from A and B, or you have some community product that you want to maximize. You just have to observe N1 and N2. And for example, here, this product P, the reinforcement learning controller can maximize the product and reduce, use up all the available food source or feedstock, in this case, glucose, and it will automatically, do this without observing the product. So that's quite useful. And there's other things that you can do with it that I think could be useful for evolution experiments. You could do directed evolution and other things. Okay, so the next part here, I want to talk about optimal experimental design. So this is the idea that we don't collect data passively, okay? Especially if you've got a chemostat or something. I mean, maybe it's a bit harder in vivo, but we can potentially interact with our experiments. We can perturb them. So the question is what's the optimal perturbation in order to extract the most information from your system? That's optimal experimental design. So you might have some experimental input that you can add, and then you take some observations. So we adapted the reinforcement learning for optimal experimental design. And here's the idea, is that you have an idea of your model for the system. It might be generalized Lotka-Volterra, might be a mechanistic model. And you want to parameterize that model. So what you do is you train, prior to doing the experiment, you train the reinforcement learning agent to maximize the reward, which is the determinant of the Fisher information matrix. That's what we started with. You could do different things. So then once you've trained the agent, the agent can then control your experiment and collect some data, and you do inference on that data with your model, and you're guaranteed to get, maximal information from the experiment. So it assumes a time series experiment. So again, we used the chemostat model. In this case, some of these aspects were non-identifiable. So we just had, so the measured variable was n, and then we had these control inputs, okay? And we wanted to estimate these three parameters. So here's like three different cases. So rational design, so that's where Nathan decided that was a rational experiment. So you might, in time, ramp up your two inputs to try and extract a time series that gives you most information. So the time series is here. Okay. This is a one-step ahead optimizer. So it optimizes one step ahead, as the name suggests, and it gives rise to this time series. And then the reinforcement learning doesn't look too different. There's no way of kind of saying, why has it done that? I mean, that's the thing about AI, right? You don't know why it's doing it in this complex case, but you don't really mind because you're parameterizing your mechanistic model. So you're kind of making use of the fact that you don't know what it's doing, but at the end, you'll be able to parameterize your model. And here's the experiments. So it might be a bit late in the day to go through this, but I'll try. Okay, so this allows us to train over a distribution of parameters. So that's different to traditional optimal experimental design. And the cool thing about that is that the agent adapts the experiments depending on what it thinks the parameters are as it's going along, okay? So in some sense, the agent can already work out what the parameters are going to be and change the experiments in order to maximize this information, okay? And here's some comparisons with Model Predictive Control. In this case, the Model Predictive Control knows the right model, okay, and we do pretty well. So what we're doing right now is we're using this we're applying this on communities. So here's generalized Lotka-Volterra, so you all know about this. So often the case, it's non-identifiable from your time series experiments. This allows you to alter the input such that not only is it identifiable, but you infer all those parameters, okay? I mean, this is only, this is 3 by 3, and there's this perturbation matrix, but, you've already got a lot of parameters. But the Fisher information matrix looks pretty good, actually. So it means it's identifiable and you can extract those parameters. So the idea is that if you can interact with the experiment, then you might be able to disentangle all these complex interactions. So just a little summary there. So on the community control, so this approach is quite general. It can be applied whenever there's an online measurement. And we're doing that in bioreactors at the moment. And then this experimental design allows us to build better models of these systems and to work out what's going on in them, okay? So in the last part of my talk, I've got a few minutes, I'll come back to the idea of microbiome engineering. So here's a project we're working on. Again, it's got a classic tongue twister backronym, but we just call it Pac-Man, okay? Doesn't matter what it stands for. But basically, we're looking at the tumor microbiota. So in certain tumors, you know, there are certain bugs that get in there, like E. faecalis, F. nucleatum, and you have a significantly poor prognosis due to DNA damage, reactive oxygen species, unknown mechanisms. But it's clear that there's a clinical impact, okay? So some tumors have these bugs, some don't. So what we want to do is, so what we're doing is engineering bugs to colonize the tumors, secrete bacteriocins, and kill the target. In this case, E. faecalis. So that's kind of the project. But, you know, we want to be able to understand, you know, with microbiome engineering, you want to be able to model what's going to happen when you put the bugs in there. And we've heard a lot about Volterra models. You know, this is from Kat Coit, who probably presented it this week. So there are some successful uses of Volterra models, and Babak also outlined why sometimes they're successful. But the other thing we can do is we can use machine learning. And people have already started doing this, and you know, it's not that difficult to apply a recurrent neural network to look at time series data. And in fact, Ophelia Venturelli has got a paper recently in eLife where she looked at trying to predict community dynamics from sub-communities. But essentially, recurrent neural networks, they do a kind of auto-correlated prediction. So you input the initial point, and then you derive what the dynamics are after that. So, you know, we've been developing these methods to run on microbiota data. And, you know, this is so far, this is all just method development and simulation. But I was quite surprised at how optimistic this looks. But, you know, it's simulation, I know, but I thought this would be unachievable. So with 288 time series of 10 time points, we were able to predict, you know, this is essentially like a fecal microbiota transplant. So we at this point, we put in a bunch of bugs, and we can predict the dynamics. It's not a very good plot, I know, but we can predict the dynamics of that. So that's pretty optimistic to me. I mean, we will have to collect these time series, but people are doing that right now. Obviously, there's a lot of noise in the systems and so forth, but I think that's pretty interesting that we might be able to do this within the next few years. But what we will need to do, I think, is, so this is kind of an extended Volterra model that we've been developing that incorporates, you know, metabolites and host factors. And, you know, mechanistic models are great. You can fit them on individuals in principle. You can capture heterogeneity. But as we've heard, your mechanistic model doesn't always represent what's going on in reality, and that's a big part of what people have been presenting this week. On the other hand, machine learning can capture these complex dynamics. They could capture memory in the time series, etc. So what we need is some bridge between these two, and that's what we're working on at the moment. So, for example, physics-informed neural networks, where you can kind of put some of these dynamics into the structure of the network. So it means that you don't need as much data, and you can capture some of the known and important interactions. I think we need new methods. We need new method development. Okay, so that's all I wanted to say. My conclusions. I think these interpretable machine learning and AI tools could be very useful for understanding microbial communities, and we shouldn't turn our backs on the field. And I think it will become part of the scientific method as long as we can interpret the results. So I'd just like to say thanks to the group, in particular Nathan and Alex, whose work I showed, Brian Ingalls, who's worked with us on the optimal experimental design, and thanks to you all for listening. And I'll take questions.
Thanks a lot, Chris. Yes, questions. Please.
You showed this model, this situation where you could optimize product production, right? So in order to do that, what do you need to measure on land? Just the amount of cells would suffice?
Yeah.
And you don't need actually to measure the metabolite concentration for like whatever you're producing? What are the, because like suppose you want to produce like a metabolite, you need to measure every 5 minutes how much you have that metabolite in order to optimize how much you produce, and that would be quite complicated.
Yeah, but what gets optimized is the glucose utilization. So it will maximize the two numbers of the species, the two species numbers.
So you only need the species.
Yeah, basically, yeah. So it has to be linked to the number of
You know that how much you produce.
Yeah, well, you need to know that it's in direct proportion of the two species. So if you remembered, it was 2 to 1. So it maximizes that ratio 2 to 1.
Because you need to kind of know which ratio you need.
Yeah. So you need to know the ratio, but you don't need to observe the product.
So I had a related question to this, if I can jump in. So in the experimental design case, what is the reward? I didn't quite get that.
The reward is the determinant of the Fisher information matrix.
Right.
So the smaller that, or the smaller that is, the better.
So, but it looked like you're changing continuously something, whereas I would have thought you have to finish an experiment and you design the next experiment, right?
Yeah, it is done in epochs.
Okay, yeah.
Yeah.
I see, okay. And there was another question.
So, really interesting. The problem with a lot of these things is when you sort of move outside the parameter space that you've been trained on, and things fall apart. So when you're talking about 5 reactors for 24 hours, will give you enough training data to continuously run one of these systems. Have you looked at how resilient one of these would be to a shock to take it out of the parameter space?
So it's a good question, yeah. And we'd hoped, I mean, we haven't looked at it in as much detail as I'd like, but we did model getting a mutation. So in principle, the controller should be able to adapt to getting a mutation, so that it would change the policy depending on its, if the mutation changed the growth rate. That I didn't show it, but there's an explore rate. So at the start you have a high explore rate, which means you take random moves, and then slowly you decrease that until you don't explore anymore and you just use the policy. But if you left an explorer, a non-zero explorer, you could potentially capture these effects. But we studied it a bit, and it needs more work to look at. But yeah, but if your parameters are outside the range completely, then yeah, I'm not sure what it would do.
There was one question here.
Well, we train the agent to maximize the redundant of the information matrix, but that depends on the parameters of the model, which you don't know yet. Like you're optimizing the experiment to find these parameters.
That's a very good point. And in fact, traditional optimal experimental design approaches, in a way, assume you already know the parameters and you update them. But here, because we do it we optimize over the distribution, we optimize over all parameter combinations. And that's why, in some sense, the agent has some relation between the parameter and the observed time series, and it can update the experiment. So if you move into it, if it turns out that the parameter is in a particular region, then it will generate time series of perturbations relevant for that part. Yeah.
Thanks a lot. I was also going in that direction. So did I get it right that the output layer of that network are the actual parameters of the mechanistic model?
The output layer. No, the output layer is not. It's the actions. So it's the perturbations that you're adding is the output layer.
And then another unrelated question. So how do you think you implement in practice an online measurement of community composition coupled to a bioreactor?
So there are, the Chi Bio, which is from Harrison Steele at Oxford, that you can measure OD online. You can also measure fluorescence as well. So there are ways, and if you've got lots of money, then you can do, infrared spectroscopy and things like that. So there are ways to do it online, but yeah, it's not straightforward.
We were hearing before, and you can couple it to a flow cytometry.
Have you seen my grant application? Yeah, there is a way that there's a bit of kit, I can't remember what it's called, but it can sample bioreactors and it can pipe it into a flow cytometer. Although it doesn't work with our flow cytometer, but if you've got a BD one, it'll work with it. And you can do flow online. Yeah. Have you got that? Yeah.
Yeah, the online. You just use pumps.
Okay, you managed to do it.
Make like a little hole in the bottom of the photogrammetry tube and you just pump it.
Okay.
And then once it's done, you pump it out. Yeah, you can do some.
Okay.
Some engine.
So there's
Use some tape and then everything works.
there is a commercial platform, but
Yeah.
I'm sure your approach works just as well. So if you can measure those things, then you can do some cool things.
All right, any more questions? Yes? Alex?
Might be a little like track, because I haven't thought about this for a long time. But like, I'm curious about how close this stuff is to a mature technology. So maybe you spent a lot of time trying to get all the little tuning things right in the architecture of the neural network, or maybe there was some package off the shelf and you just popped it in, and if you know the science, then you're ready to go. How much of so you say like, oh, let's adopt AI, let's not shun it. How much extra burden at this point in time is it to do the adoption of AI?
I should point out that I didn't do any of the programming. PyTorch or what, you can. I'm teaching PyTorch. I mean, that's how easy it is. So you can set these networks. So it's still a dark art as to how many layers you need, how many layers, how many nodes per layer, neurons per layer, things like that. But generally speaking, the challenge wasn't the network. You know, the network was fairly straightforward in both these cases. Well, actually, that's not true. In the optimal, so in the control, we just use Q-learning, okay? And that's your basic entry-level reinforcement learning. Then we iterated it and we did something called fitted Q-learning, which is slightly more advanced. In the optimal experimental design, we had to have a reinforcement learning approach that is continuous, so that was more tricky. And I should say there was a recurrent neural network inside that as well, so that was a bit harder. But still, you know, it's doable in standard PyTorch, if you know what you're doing.
All right, can I chip in a possibly silly or philosophical, whichever? Is there going to be then a reverse engineering of the strategies of the thing actually uses? Because you're always saying, like, you can't say what it's doing or why it is doing it.
Well, in the first, remember when I showed that policy plot, the simple one, and you could see which auxotrophs, which amino acids, that, you know, we reversed engineered that, but obviously it becomes more difficult. But for optimal experimental design, all right, I'll balance the philosophy back to, do you need to, you know, if you parameterize your model, do you need to understand why it proposed those experiments? But yeah, it's challenging. I mean, that's the thing you lose with neural networks, right?
On that philosophical note, let's thanks